Welcome back. So, we looked at in the last lecture some of the important mechanisms in biological chemistry. So, we are going to continue with the same theme and look further into some of the mechanisms that are very interesting and relevant to in biological chemistry. So, just to put this in perspective, we looked at this very large picture of how metabolism occurs, and we looked at some key molecules such as pyruvic acid and how pyruvic acid actually can plays very important roles in transferring groups. And we looked at how alpha keto acids are actually, such as pyruvic acid, is actually in equilibrium with the corresponding amino acid, and this transfer that can occur is catalyzed by aminotransferases. And we also looked at enol equivalents, and because it is not possible to generate an enolate, or it is unlikely that an enolate will be generated inside the cell, and so the cell or nature takes care of this by making very interesting enol equivalents. So, one of the enol equivalents that we looked at was phosphoenol pyruvate, and so on. So, now in today's class we look at acyl anion equivalents. So, acyl anions are very important in many reactions that we will that we conduct in the organic chemistry lab, and the way it happens in nature is through thiamine pyrophosphate. So, the structure of thiamine pyrophosphate is shown here, and what the interesting part of the structure is that it contains a thiazole ring, okay, and this thiazole ring is actually positively charged because this nitrogen has 4 bonds to it, and so this provides a very interesting chemistry that we will look at soon. But this thiamine pyrophosphate, as the name suggests, contains pyrophosphate here as shown here, and it also contains the pyrimidine ring over here. And so, this together this molecule plays a very important role in nature. So, the key part of this molecule is the thiazolium salt, which is shown here in the middle. So, here is a proton or a hydrogen which is next to this positively charged nitrogen species, which can be deprotonated and form a carbanion. So, to put this in perspective, if you imagine a ketone which has an alpha hydrogen next to it, if you add a strong base, then you would expect that a carbanion is produced. Okay. This carbanion is in resonance with the enolate form. So, here the proton between the nitrogen sulfur atoms, and it can be removed, and it forms what is known as an ylide. Okay. So, ylide is something that we have encountered previously in organic chemistry courses when we are looking at a Wittig reaction. So, this ylide is basically an anion which is in the sp2 orbital, and it adds to the reactive carbonyl group of pyruvate. So, once you produce this carbanion can then react with the carbonyl of pyruvate to form a carbon-carbon bond. And now, the carboxylate ion can be lost from the form of pyruvate as positively charged imine in the thiamine molecule, which provides a perfect electron sink to take away the electrons. So, recall we had a very similar situation in the positively charged NAD plus, right, where you had a positively charged nitrogen which was the electron sink. So, similarly here you can push electrons from the O minus to produce carbon dioxide. This carbon-carbon bond is broken, and you end up with this nitrogen as the electron sink, and you produce nitrogen with 3 bonds which is neutral. Okay. So, therefore, the formation of carbon dioxide forms a very important driving force in this reaction. This new intermediate contains a new and strange carbon-carbon double bond. Okay. So, this carbon-carbon double bond has on it an oxygen, a nitrogen, and a sulfur, 3 electronegative atoms sitting on it. Okay. So, this makes this double bond quite electron rich, and at this point this electron rich double bond can react with disulfide functionality of lipoic acid. Okay. So, to just recall or to understand what lipoic acid structure is shown here, which has this disulfide bond. And so, this C double bond C, which is electron rich, can react with a disulfide and produce a carbon sulfur bond, and it breaks the disulfide to produce a thiol. Now, if you see here, this thiamine regained its positive charge. So, now this thiamine, which has a positive charge on it, can again react to produce the carbanion, which is originally reacting with pyruvic acid. So, here is how that mechanism happens. So, you can have a basic residue in an amino acid to come and pick up this proton from this alcohol, which then generates the ketone as shown here, and this carbon carbon bond breaks and regenerates the carbanion. So, this is the ylidd of thiamine, which functions in a very crucial way to transfer an acetyl group. So, the product is a thiol ester, and so it can exchange with a COA-SH in the simple ester exchange reaction. So, you see here that COA-SH, as we have looked at in previously, can come and react with this acetyl group and then regenerate this dithiol intermediate, which is basically the reduced form of lipoic acid. Now, this acetyl CoA species is produced, and the dithiol can be oxidized to produce the disulfide, okay, and which is basically regenerates the lipoic acid. And this oxidation is very facile, and it would occur in air, but in nature it is carried out by another cofactor known as FAD, which is flavine adenine dinucleotide, which is a very close relative of NAD+. So, thiamine pyrophosphate also catalyzes reactions of alpha keto acids other than pyruvic acid. So, if you take pyruvate and react it in the presence of thiamine pyrophosphate and NADPH, you can actually form valine and isoleucine. Now, let us look at this remarkable chemistry in the biosynthesis of branch chain amino acids valine and isoleucine. So, the remarkable aspect of this chemistry is that it involves a 1,2 alkyl shift, which is resembling a pinocol like rearrangement. So, in order for us to understand a pinocol like rearrangement, let us go back and look a little bit in detail what this rearrangement is. So, a pinocol is basically this group here, which is shown here, where there is a hydroxyl group on the 2 and 3 positions. So, there are flanking hydroxyl groups, which in the presence of an acid such as sulfuric acid gives you what is known as a pinacolone, okay, which is basically a ketone. So, where if you number the carbons here, you have 1, 2, 3 and 4. So, the carbon here is 1, 2, 3 and 4, and so you form a ketone on carbon 2, and the original alcohol that was present in carbon 2 has now gone, and carbon 3, which has an alcohol, is also now going to be occupied by a methyl group. So, this is known as the pinacolone rearrangement, and now we should look at the mechanism of this reaction. So, in the presence of acid, it is quite likely that this alcohol will undergo protonation. So, once it undergoes protonation, it is going to perhaps lose water to generate a carbocation. Okay. So, this carbocation, as we know, is going to have an empty p orbital, which can facilitate this rearrangement, or which can act as a place where this new carbon carbon bond is going to form. So, what happens is that the lone pair on the oxygen then comes in, and this carbon methyl bond breaks and forms a new carbon carbon bond with the adjacent carbon, and you form a protonated carbonyl species as shown here. And loss of this proton gives you the ketone, which is pinacolone. So, carbocations are typically rearranged by alkyl shifts, but this carbocation is already in the tertiary form, and so there is no need for it to rearrange. So, the reason why this rearranges is that the lone pair on oxygen plays an important role. So, since the lone pair on oxygen can now move in and form a stable ketone, this constitutes the driving force for this reaction. Also, oxygen is very good at stabilizing a positive charge on an adjacent atom, and somewhat less good at stabilizing a positive charge 2 carbons away. So, by rearranging, the first formed carbocation, which is here, okay, gets the positive charge into a position where oxygen can stabilize it and loss of a proton from oxygen then gives a stable ketone. So, let us look at an example here of the spinacol-pinacalone rearrangement, and here is the pinacol type structure with two 5-membered rings, and this rearranges and gives you this product here. So, the mechanism that we would propose would be protonation of this OH to give you OH2 plus, and subsequently the rearrangement or the shift of this carbon-carbon bond here will give you a spirocyclic system. So, a very useful tip in solving such rearrangement mechanisms is to number the carbons. So, if we are able to number the carbons correctly, then we can keep track of which carbon ends up where. So, if you see that this is carbon number 1 and this is carbon number 1, if you keep track of it, then you know that the carbon number 1, which has an alcohol initially, ends up with a ketone, and the bond that forms is between carbon 2 and carbon 6, which is shown here. Okay. So, now just to recall or put it in perspective of what we are looking at in thiamine. So, thiamine forms a carbanion, which is shown here, and this carbanion reacts with pyruvate to give you this alcohol here. The carboxylate then undergoes decarboxylation to produce this enol shown here. And as we discussed, this enol is a very interesting enol because it has 3 electronegative atoms on it, and it can then serve to do very interesting reactions. So, the sequence of reactions starts now. So, this enol reacts with the ketone or the alpha keto carboxylic acid, and then it forms an intermediate which is exactly identical to the pinacol type reaction. So, here the decomposition of this product with the release of the thiazolium ylide can occur. It also releases the product of a coupling reaction between 2 keto acids, that is it forms a 1-hydroxy 2-keto acid. So, the thiazolium ylide is now free to catalyze the next round of reactions. So, the hydroxy keto acid is now primed for the rearrangement. So, the migration of the R group is pushed by the removal of a proton from hydroxyl group. Okay. So, you can think about this enzyme with a basic residue here coming in, picking up this proton, and then this R moves here to and reacts with the ketone and gives you the product. Notice that the R group, which is methyl or ethyl, migrates in preference to the carboxylic acid. So, usually the group that is better able to accommodate the positive charge migrates. So, here again the enzyme plays a major role in determining migration. So, control in this reaction is likely exerted stereoelectronically by the enzyme, as it was done in the pyridoxal reactions which we discussed previously. Since the carbon R bond is held parallel to the p orbitals of the ketone, R migration occurs. But if the carboxylic acid were to be held parallel to the p orbital, then decarboxylation would occur. So, which is what we saw in the previous case in the case of the NAD reaction. So, a simple reduction with NADPH converts the ketone to an alcohol and prepares the way for a second rearrangement. Okay. So, when this reduction occurs, then you end up with the dihydroxy compound as shown here. The second rearrangement is even more like a pinacol rearrangement because the starting material is a 1,2-diol. So, the tertiary alcohol is protonated and leaves, and again the carboxylic acid does not migrate even though the alternative is merely hydride. Okay. So, here you again have the shift of this carbon hydrogen hydride here to kick out hydroxide ion to give you the product as shown here. Finally, a pyridoxal transamination converts the 2 keto acids stereospecifically to the corresponding amino acids. So, where R is methyl, it becomes valine, and when R is ethyl, it becomes isoleucine. So, the donor amino acid is probably glutamate, and it usually is in the amino acid synthesis. So, this enzyme catalyzed pinacol rearrangement gives you the final product, which is valine or isoleucine. The next topic that we will look at is carrying carbon dioxide. So, we would not expect gaseous carbon dioxide to be available inside a cell. Instead, CO2 is carried out as a covalent molecule by another coenzyme known as biotin. So, here is the structure of biotin, and it has this thioether, which is in a 5-membered ring, and it also has a urea functional group on it. And this can react with carbon dioxide to produce a species such as this. So, biotin has 2 fused 5-membered ring heterocyclic rings, and the lower is a cyclic sulfide and has a long side chain in ending up in a carboxylic acid. And so, this helps with attaching itself to lysine residues of a protein. The upper ring is a urea, and it has a carbonyl group that is flanked by 2 nitrogens. It is this ring that reversibly captures CO2 on the nitrogen atom opposite the long side chain. The attachment of the enzyme as a lysine amide gives it an exceptionally long flexible chain and allows it to deliver CO2 whenever it is needed. One of the important points at which CO2 enters as a reagent carried by biotin is in the fatty acid biosynthesis, where CO2 is transferred from to the enol of acetyl-CoA. We have already looked at the major aspects of the structure and reaction of acetyl-CoA. But here a magnesium ion is also required, and one can imagine the reaction as a nucleophilic attack of the enol on the magnesium salt of carboxybiotin. So, what we would expect is that magnesium would form some sort of a chelating arrangement as shown here, and then we just looked at acetyl-CoA being an excellent enol transferring group, and so you can form this is the enol of acetyl-CoA, which then reacts with this carbon dioxide and produces this type of a product. Now, let us look at a very important pathway, which is the shikimic acid pathway. This pathway is responsible for the biosynthesis of a large number of compounds, and it is very important in plants. Most importantly for us is the biosynthesis of aromatic amino acids, which are phenylalanine, tyrosine and tryptophan, whose structures are shown here. These are classified as essential amino acids, and we have to have them in our diet as we cannot make them ourselves. So, we get them from plants and microorganisms. So, how do plants make these aromatic rings? So, a clue to this structure, to this chemistry comes from the structure of caffeil quinaic acid, which is a compound that is present in instant coffee in some quantity. It is about 13% of soluble solids from coffee beans. So, here is the structure of the caffeil quinaic acid. It has this very nice sugar unit here, or a carbo sugar unit here, and it has this dihydroxy benzene ring, which is attached to alpha beta unsaturated ester. So, this ester has 2 6-membered rings, one aromatic and one rather like a sugar alcohol. So, you can imagine that dehydration, that is losing 3 molecules of water from this species, is going to give you a caffeic acid. It is now known that both rings come from the same intermediate, which is shikimic acid, whose structure is shown here. So, we shall now look at the biosynthesis of shikimic acid, or conversion of shikimic acid to an aromatic ring. So, the key intermediate has given its name to nature's general route to aromatic compounds and many others related 6-membered rings, which is called the shikimic acid pathway. This pathway contains some of the most interesting reactions, especially from a chemist standpoint in biology. It starts with a very interesting aldol reaction between a phosphoenol pyruvate, which we have looked at previously, as the nucleophilic enol component, and a C4 sugar erythrose 4 phosphate as the electrophilic aldehyde. So, here is the electrophilic aldehyde, which comes from erythrose 4 phosphate, and here is the phosphoenol pyruvate equivalent. And now you can imagine an aldol reaction that occurs, which gives you this product. And now this can subsequently break to give, or can be hydrolyzed by water to give you inorganic phosphate and give you the ketone. So, once it gives you this ketone, this intermediate has the right number of carbons for shikimic acid, and the next stage is a cyclization reaction. So, here the cyclization reaction gives you this intermediate as shown here. Now, if we redraw this C7 alpha keto acid in the right shape for cyclization, we can see what is needed. So, here is the bond that we need to form, and this can happen in the following manner. This reaction looks like an aldol reaction too, but there is an obvious route to the required enol by elimination of the phosphate. This would require removal of a proton, which is here, from this molecule, but this is not at all acidic, right. So, you can imagine that it can go through an E2 process wherein this proton is lost and give you this enol, which then can react with the ketone. But since this hydrogen is not at all acidic, nature adopts a very interesting route to carry out this reaction. Let us look at that now. So, the problem is avoided if the hydroxyl group at C5 is first oxidized to the ketone. So, we have already looked at NAD plus can be the oxidant. So, what we are doing here is that this hydrogen shown here, which was previously not acidic, is now adjacent to a ketone, and now this pushes down the pKa substantially and makes it acidic enough. Now, if it undergoes enolization, the enolization is going to give you an enol such as this, which can undergo E1Cb type elimination reaction to kick out phosphate and give you this product. Subsequently, reduction by NADH perhaps is going to give you back the alcohol here, which then undergoes aldol reaction to give you the desired product. The product is actually dihydroquinonic acid and is an intermediate on the way to shikimic acid. It is also in equilibrium with quinoic acid, which is not an intermediate on the pathway, but which appears on some natural products like coffee ester caffeil quinic acid. The route to shikimic acid in plants involves, as the final steps, the dehydration of dihydroquinonic acid and then reduction of the carbonyl group. Doing the reactions this way around means that the dehydration can be an E1Cb, which is much preferred under biological conditions. So, what we would expect is the enolization here of this ketone to give you the corresponding enol, which then if you do an E1Cb reaction can eliminate this hydroxide to give you the dihydroshikimic acid. The final reduction uses NADPH as the reagent and is of course totally stereoselective, as we have looked at previously, with the hydride coming in from the top face of the green ketone as drawn here. Okay. So, then after this stereoselective reaction, you get this alcohol here, which is basically shikimic acid. Of course, we are here only halfway and we have another half to carry out. Here, the first step is a chemoselective phosphorylation of one of the 3 hydroxyl groups by ATP. As it happens, the hydroxyl group that has just been formed by reduction of the ketone. So, the first step here is the phosphorylation over here, and this step prepares the hydroxyl group for a later elimination. Next, a second molecule of phosphoenol pyruvate appears and adds to the hydroxyl group at the other side of the molecule. So, here is the other side of the molecule. Here phosphoenol pyruvate comes and adds here and gives you this product. This is the phosphoenol pyruvate in its enol ether role, okay, and forms an acetal under acidic catalysis. The reaction occurs with retention of stereochemistry. So, we know that the hydroxyl group acts as a nucleophile and that the ring hydroxyl group is not broken. Now, a very interesting 1,4 elimination occurs, and this is known to be a syn elimination on the enzyme. So, here this 1,4 elimination occurs in the following manner. So, the enzyme, basic residue of the enzyme comes in, attacks here, moves this electron, moves here, this bond moves here and kicks out phosphate. And so, this gives us chorismic acid, which has a 1,3-diene. Right. Of course, in the laboratory when we do this reaction, this elimination can be either syn or anti, but since this is happening enzymatically, there is a stereochemistry which is very specific stereochemical outcome is expected. The product here is chorismic acid, and this undergoes the most interesting step of all, which is basically a 3,3 sigma-tropic rearrangement. So, this 3,3 sigma-tropic rearrangement is something that some of you may be exposed to in your prior classes, and sigma-tropic rearrangements are basically neutral reactions which occur in a concerted manner. We will not have time to go through this mechanism, but what happens here is that you form a new carbon-carbon bond, which then as shown here, and then you break this carbon-carbon double bond, and this carbon-oxygen bond is broken, and you form a new C double bond O. Okay. So, this occurs in a 3,3 sigmatropic manner, and it is called a suprafacial rearrangement. So, the most favorable conformation, if we draw out the stereochemistry, has the substituents in the pseudo-equatorial position, okay, so which is shown here, right. But in the pseudo-equatorial position, the rearrangement cannot take place because it is not in the right orientation. So, it has to first flip and form the diaxial conformation, which is shown here, which is known as the pseudo-axial conformation, and then we can imagine a chair-like transition state that occurs as shown here, which is going to give the product, and all the required orbitals are correctly aligned in this conformation, and therefore, this is favored. These reactions occur well without an enzyme, but the enzyme accelerates this by about a factor of 1,000,000 increase in rate. So, there is no acid or base catalysis, but we may suppose that the enzyme binds the transition state better than it binds the starting materials. So, if we were to design a molecule which has a which looks similar to the transition state, which we have looked at previously as transition state analogues, then this compound can go and inhibit the enzyme. And indeed, when a molecule that was synthesized which resembles the transition state as shown here, this molecule can inhibit this enzyme. So, in this whole process, we have arrived at pre-phenic acid, which as its name suggests is the last compound before the aromatic compounds are formed. So, this is the end of the shikimic acid pathway, and the final stages of formation of phenylalanine and tyrosine start with aromatization. So, aromatization occurs by a decarboxylation followed by pushing of electrons to give you to kick out hydroxide ion. So, the way this would occur is that you can imagine that CO2 would be lost by this from this carboxylate ion, and then this pushes electrons in this manner to give you phenyl pyruvic acid. And we know that phenyl pyruvic acid, which is an alpha keto acid, can exist in equilibrium with its corresponding amino acid, which is phenylalanine, and this transamination is mediated by pyridoxal. The route to tyrosine requires a preliminary oxidation followed by a decarboxylation. So, here this prephenate will have to undergo oxidation to give you this ketone, and then for subsequent decarboxylation will give you the tyrosine residue. And of course, this is it gives you the alpha keto form, which then is going to exist in its equilibrium with the corresponding amino acid, which is tyrosine. The next topic that we are going to look at is how oxygen is carried inside the cell. Hemoglobin carries oxygen, which is as an iron 2 complex, and this is very important because a number of biological oxidations, which we shall look at shortly, is mediated by heme containing enzymes. And human metabolism depends on oxidation and on getting oxygen, which makes up about 20% of the atmosphere inside cells. The oxygen transporter from atmosphere is hemoglobin, and the reactive part of this molecule is the porphyrin ring, which is shown here. So, porphyrin is actually aromatic because it contains 18 electrons, and if you apply the 4n plus 2 rule, then when n equals 4, you get 4n plus 2 to be 18, and so therefore, this is an aromatic system. And these are symmetrical porphyrins are easily made from a pyrrole and an aldehyde. So, if you react this pyrrole with an aldehyde, you get this kind of a porphyrin. Then this porphyrin contains a hole in the middle, which is just the right size to take a divalent transition metal ion. Okay. So, in the first transition series, such as zinc, which is forms zinc porphyrin, and once which is inside this complex, it is extremely stable. It is very difficult to get it out. So, two of the nitrogen atoms form normal covalent bonds, and the other two donate their lone pairs to make 4 ligands around the metal. The complex zinc atom is square planar and still has 2 vacant sites above and below, which can coordinate with certain ligands. And so these ligands can be either ammonia or water or other ligands. The porphyrin part of hemoglobin is called heme, and it is an iron 2 complex. It is unsymmetrically substituted with a carboxylic acid chain on one side and vinyl groups on the other side. Heme is bound to proteins to make hemoglobin, so in the blood, or it forms myoglobin in the muscle. The hydrophilic carboxylate groups stick out into the surrounding medium, while the majority of the molecule is embedded in a hydrophobic cleft in the protein, which is lined with amino acids such as leucine and valine. Okay. The octahedral coordination sphere of iron 2 is completed with a histidine residue as shown here and an oxygen molecule which is shown here. The oxygen complex can also be drawn as an Fe3 complex of the oxyanion. So, here is the oxygen complex, here is the resonance form of it, and it can be drawn as an oxyanion. Oxygen molecules are transferred from hemoglobin to other hemes such as P450, which we shall encounter very shortly, and to a wide range of other oxidizing agents. Almost any molecule that we ingest that is not a nutrient, which for example a drug molecule, is oxidized. And the way this oxidation occurs is that this X is the drug molecule, which can get oxidized to give you X double bond O, which leaves this molecule behind.